Let's start the next session. Synthetic constructions. And I just want to say that at the very root of synthetic biology is the ability to synthesize genes and then assemble them into either pathways or chromosomes. And so this session focuses on some of the methodology and types of constructions that one might do. Our first speaker is Clyde Hutchison. He's from the J. Craig Venter Institute in La Jolla, talking about genome construction.
Good morning. I'd like to thank the organizers for the chance to tell you all about the work going on in our synthetic biology group at the Venter Institute. And Ham talked last summer in Zurich, about our project to look for the minimal set of genes to direct a living cell by the approach of building a synthetic genome and installing it in the cell. And I want to give you an update on that project. This is really a team effort, the synthetic biology group. Here it is. This picture is a little out of date, a fairly diverse set of humans. And we were all in this building in Rockville. It's apparently a picture taken on the weekend, and there's Ham's white Cadillac with the powerful North Star engine. And a little after this picture was taken, some of us moved to La Jolla. And here's the building we're located in La Jolla. And it's a real team effort. I'll put the names up at the end. I'll mention some of the stars as we go along. I'm afraid I'm in the 10%, or is it the 90% or maybe 99% of people who don't quite understand what synthetic biology is. And so rather than getting into the way of making a definition, we like to talk about what we're doing as synthetic genomics, which has to do with the chemical synthesis and building up from scratch at a chemical level genomes or chromosomes or pathways, but using chemical DNA synthesis to build genetic material. And the current objective of this work is to make bacterial chromosomes, which could be used to produce new cells that have useful properties or make useful new products. And to put this in just a little bit of historical perspective, Khurana, of course, is the father of DNA synthesis and gene synthesis, and he developed methods for making he developed the basic methods for synthesis of oligonucleotides and built the first synthetic gene, a tRNA gene, in the 70s. And since that time, methods have steadily improved with the introduction of automated synthesizers and so forth. And in the last few years, it's become clear that it's possible to synthesize any viral genome that you want. And some examples are polio, coronavirus, not SARS virus. All these things synthesize, flu, so forth. And the cost for DNA synthesis has dropped dramatically over time. And as I'll discuss today, it's now possible to synthesize whole bacterial genomes, at least small ones. And our premise here is that this analogy is pretty close to correct. That is, that the genome of a cell can be considered as the operating system, and the cytoplasm is the hardware. So that if the DNA is the operating system, and the cytoplasm provides the ribosomes, enzymes, and so forth necessary to express the genome. And either one separately is not alive. And this is a fairly good analogy. I mean, some of the details are correct, such as you can't always expect any operating system to work on any arbitrarily chosen hardware. Okay, we got interested in building synthetic genomes from the perspective of wanting to understand what the basic genes are for life. And so we like to talk about something we call a minimal cell. And this would be a cell in which all the genes are essential. Disrupting any one of the genes would lead to a non-living cell. And we are interested in this under the most permissive laboratory conditions that we can generate. And why be interested in that particular condition? It's because in those conditions, all the fundamental genes that have to do with genome duplication, expression of the genetic material, all those have to be able to function, but genes necessary for things like interaction with hosts or interaction with the environment, which are special to each situation, don't need to be there. And so this gives us an idea of what the minimal set of genes are that a cell can have. And we're interested in this because hopefully one day it'll be possible to model a cell's behavior on the computer and determine the effects of environmental variations and so forth on the computer model and be able to make predictions about how to design genomes. Also, if we had a minimal possibility, it could be made more useful by adding things to it. And so we asked the question, can we chemically synthesize a minimal genome? And with the definition I've described. And this sounds very simple, but if you think hard about this definition, it's really pretty complicated. But the basic idea is, can we make cells that are considerably simpler than any known living cell? So it makes sense in this problem to start with a cell that has a natural genome that is small, and started with Mycoplasma genitalium, which has the smallest genome of any cell that has been grown in pure culture. And it has about 485 protein coding genes and 43 RNA genes. In fact, that's exactly what it has in the current annotation. And the question is, how many of these genes are essential, and what is needed to run a living cell? So here's a picture of the cell taken in the electron microscope. And you see, even though it's very simple, it has a defined structure. It has a tip organelle that has adhesins that allow it to attach to its host cell in the natural environment. And those genes are here when the cell's grown in pure culture, and that's an example of a kind of gene that you think would be dispensable in the laboratory. And here's another picture of the organism. One of the problems with it is it's a very slow-growing organism. So after 3 weeks, we have colonies here. We have colonies that are only 10th of a meter. So it's but it grows on a complex medium, and it's slow to work with is the downside. So we spent a number of years trying to see whether, to get an approximation to what is the minimal set of genes out of the 500 naturally occurring genes. And several approaches have been used that I'll just summarize in this one slide. there are about 300 minimum gene sets been estimated at around 300 based on comparative genomics approaches. And this started off by comparing the genomes that were available, Mycoplasma genitalium and Haemophilus, but the approach is much more interesting now that we have about 15 different Mycoplasma genome sequences. Another kind of approach that's been taken is an experimental approach using gene knockouts. And we have been able to knock out about 100 of the annotated genes in Mycoplasma genitalium individually, one at a time. And this then would lead to the estimate that there are 385 essential proteins. And of these proteins that appear to be essential, about 100 don't have a clearly defined function. That's, of course, one of the problems plaguing genomics in general, is the plethora of undefined genes. So we have here a situation where we know there are a lot of genes that are dispensable, but we don't know how many of them could be removed simultaneously, because our knockout procedure just allows us to look at single gene knockouts. So the approach we've decided to take is to try to construct the genome and put it in a cell, and do the construction in a way that will allow us to leave out genes and test specific hypothetical sets of genes to see if they direct a living cell. And this, I said I wouldn't talk about the stock market, but that cross out was really meant to refer to inflation, not anything about the strength of the dollar. So the general approach we've been trying to take is to build up the genome starting from oligos, which are, regular oligos made on a DNA synthesizer, sort of the order of 60 nucleotides long, assembled into subsections. We had 101 subsections. Then build those into a genome, and then try to take that genome and replace the genome of a recipient cell to produce a new cell directed by the synthetic genome. And to do this, we had the following considerations. First thing is we had to know that the sequence that we were starting with is right and will direct a living cell. So to be sure it would direct the living cell, the only thing we could do reasonably was synthesize the natural chromosome sequence, because we know it works. We had to make sure the sequence is correct by resequencing the genome. When we did that, we found about 30 differences from the original sequence, which we like to think of as errors in the original sequence. It's irrelevant whether there are errors or not. We looked at all of these 30 positions carefully, and we have a sequence that clearly that is the sequence of the strain we have in the laboratory today, and so clearly directs a living cell. And we believe that sequence is absolutely accurate. Now, we wanted to do something to identify the DNA. We added some sequences that we've called watermark sequences that are not in the natural element, but we put them in at places where we know the genome can tolerate insertions, and we put them in a way that they cannot be expressed at all as protein. Then we divided the genome into about 101 cassettes, as we call them, that overlap by a little bit. usually about 80 nucleotide overlap between adjacent cassettes. And then the plan was to assemble these in stages to make the complete genome. And if we can do this and get that genome to work, only then will we begin to try to minimize the genome. So we could have, in fact, we wanted to build all the DNA ourselves from synthetic oligonucleotides. However, making genes and making pieces of DNA several kilobases in length has become a commodity, and really the challenges that we face all lay in assembling pieces several kb into the larger segments and the whole genome. So I think one of Craig's big insights in this is that we should just order the cassettes from commercial firms, and we got them from several companies, including GeneArt, who's here, and Blue Heron actually made most of them. DNA 2.0 made some. And as I said, the difficulties lay in assembling these things. Here's just a layout of the design. The cassettes are these 101 things. The genes are shown here. Places that tolerate transposon insertions are marked. RNA genes are here. And this is the scheme that we adopted initially for assembling the genome. So we have 101 cassettes, and the idea was to join these in groups of about 4 to make 25 assemblies, each of which is about 24 kilobases. Then put those together in 3 at a time into 72 kilobase assemblies, put 2 of those together to make quarter genomes, and then build half genomes and the whole genome. That was the plan. And here is the scheme that we adopted to do this. Dan Gibson, who's here at the meeting, developed really robust procedures for assembling these pieces using the homology at the overlaps between adjacent pieces. So here are 4 of the cassettes, and to assemble those into a what we call an A assembly, a 24 kb piece, the ends were chewed back with a 3' exonuclease. In the published work, it's a T4 DNA polymerase with no triphosphates is the 3' exo. Then the overlaps will anneal, and they can be repaired with DNA polymerase and ligase. And that will assemble the 4 pieces together. However, if you want to clone this in E. coli, you can assemble those 4 pieces with a back vector that's been amplified with 2 primers that have homology at the ends of this assembly, so that if that's added to the reaction with the 4 cassettes, then these assemble, the ends of the back assemble here to form the complete molecule, which can be directly cloned into E. coli. And there are sites, not one sites, which don't occur anywhere in the genome that can be used to release the DNA for further assembly steps. Okay, there's Dan. So you'll recognize him when you see him around. So this just shows examples of assembly reactions to produce an A series, a B series, a 72 kilobase reaction, and here a quarter genome. And there are various products, but you see visible bands at the positions expected for the full assemblies. And after those are cloned in E. coli, you isolate the backs and cleave the insert out with knot 1, then you get clear bands of just the right size for all the assembly. So this, as I said, works very robustly. And so we followed that procedure and did quite well till we got to quarter molecules. And I should say all the things with the bold black boxes were things that were sequenced and had exactly the design sequence. So in getting to this quarter molecule stage, there were a couple of places where we had to backtrack and make things again. But it was surprisingly trouble-free. And then at the 4 quarter molecule stage, we sort of reached an impasse. We spent quite a lot of time trying to build these half genomes in E. coli without success. And here's someone else who's at the meeting, Gwyn Benders. And Gwyn decided to look at assembling the pieces using yeast recombination. And here's the construction that gave the complete synthetic genome in yeast. She took the 4 quarter molecules, cleaved one of them at a unique restriction site, designed primers for a yeast vector, and these primers overlap the sequence on the either side of the unique restriction site in the genome. And so when these 6 pieces are put together and just put into yeast without any enzymatic treatment, yeast recombination put them together. We, of course, had to screen a number of colonies by using PCR pairs all around the genome and identify likely candidates. But here is a clone that's called M. genitalium JCVI 1.0, and when you cleave that with knot 1 to release from the vector, you get a discrete band at 182 kb, which is the complete synthetic genome. And that genome was sequenced from yeast, and it's right. I mean, we think it's absolutely the design sequence. It was sequenced by Sanger sequencing to depth of, I forget, greater than 10, and fully confirmed sequence. And now Ham talked at Zurich last year, and he talked mostly about work on putting genomes into cells, and we had not yet got the complete genome synthesized. So since that, this work, the last step has been done in the synthesis, and the work has been published. And here's some pictures of the genome just floating around in water with cyber gold. And these are, pictures at intervals of roughly 100 milliseconds. You see them bouncing around and sometimes supercoiling and uncoiling. I picked this one because it's clearly a circle over some period of time. And that was fine. This process took us a few years to do, and we particularly were held up at this stage. So Dan and Gwyn became interested in the question of really, could we push the yeast assembly further to eliminate some of these steps? We spent perhaps a year or so on these steps. And in fact, it's possible to take these 25 A-series assemblies, just mix them together. These aren't exactly the same ones as in the paper. One of the assemblies has a vector in it. Take all 25 of these and present them to yeast, and again, you have to screen a few clones, but you can get the fully assembled genome. And I just edited this slide. That's being presented as a poster by Dan and Gwen if you want to get details on it. And apparently this poster is bigger than life, and so it doesn't fit in the space where it was originally assigned. So that's why I'm saying it's going to be at positions 189 and 90, and I assume there'll also be a sign at the listed location for it saying where it's moved. And not to show everything that's in the poster, but just give you some idea of what's going on. The way that this was done was to pick a number of colonies carrying the His marker that's on the yeast vector, and 10 colonies were analyzed, and we have PCR markers shown by the colored lines that are spaced all around the genome and in every one of the cassettes, more than one in every one of the cassettes. And in the initial screening, it turned out that clone 4 here had all of one set of the primers, and when it was screened with all 4 sets of the primers, all of these, it has all of the sequences at the primer locations, and then if you isolate the whole genome from yeast and cleave it with, in this case, EAG1, which has a single site on the genome, then that releases this 592, I think, kb linear form of the genome. This is doing the same thing with the parent yeast strain that doesn't carry the clone. So that experiment was done, and the analysis in a couple of weeks, and it essentially replaced more than a year of work in the assembly process. So my topic was genome construction, but I think I should update you a little bit also on what we've learned about putting genomes into cells, because we're at the stage now we have the synthetic genome, but so far have not put it into a cell. And as Ham described in Zurich last year, we built a test system for investigating putting genomes into cells, and rather than using a synthetic genome, we use genomes isolated from a cell and then put that into a recipient cell. And this, the work Ham talked about last year, was published in Science. John Glass is the leader of this transplant team, and the two of the main people involved were Carol Lartigue and Nina Alperovich. Now, in that work, we didn't use Mycoplasma genitalium because it's so slow growing, and rather we chose other Mycoplasma species. We needed to choose a Mycoplasma because Mycoplasmas have an unusual code, and we wanted the genome to be expressed and to work, so we wanted to choose a cell as recipient that had the same code as the genome we were installing. And we chose these two species, Mycoides, large colony, and Capricolum. They're goat pathogens. They have doubling times that make them almost as convenient to work with as yeast from a time perspective. They don't have well worked out genetics like yeast, of course. They have distinguishably different genome sizes that are 8% different over the homologous part of the two genomes. so they're very easy to tell apart. And without going through the details of this, the donor strain is marked with tetracycline resistance and carries also LAC-Z plus. The donor is embedded in agar plugs and treated to render it essentially free DNA as best we can judge by silver staining or mass spec. And then that DNA is mixed together with suitably prepared recipient cells. And this is very similar to plasmid transformation as done with mycoplasmas. And the transplantation is done in the presence of 5% PEG. The cells are grown out to allow expression of the TET marker and plated. Now we routinely recover about 200 events that are blue TET-resistant colonies, which it turns out have taken up the whole transplanted genome. Okay. Ham said there's only 2 speakers here. That's why I'm taking a few minutes more. Not that I, out of control or anything. Okay. There's no, in every experiment, we have 2 very important controls with no donor DNA and a control with no recipient. And by a whole number of criteria that we've published, the genotype and the phenotype of the cells, the genotype at the sequence level is pure donor, and the phenotype as judged by looking at the protein products is pure donor and no trace the recipient. We've cleanly replaced the genome of the recipient cell with that from the donor. And what we now have to do is get this system to work with our synthetic M. genitalium genome. And there are 2 levels to this. We have not so far transplanted a natural M. genitalium genome because there apparently are species-specific problems in this transplantation phenomenon. And the other problem is we need to move a genome from yeast into a mycoplasma, which also has some problems. And I'll just tell you what our thinking was when we started this. We just thought naively that PEG would somehow get the genome into the cell, it would grow and divide and segregate, and then tetracycline selection would select for the cells that happen to have taken up the genome. These are, as I said, rare events. There are about 200 such events from 10 to the 9th recipient cells. So it's hard to actually look at the early steps in the events. But, is that really how transplantation works? And one thing we began to wonder about is whether DNA restriction is involved in the process. And we also wondered, what is PEG doing in this process? Is it really a question of single cells taking up DNA or what? And we have a good handle now on the restriction question. It's clear that in our transplant experiment that worked, that DNA restriction is going on, and that the donor strain makes 5 type 2 restriction enzymes. Richard Roberts predicted those specificities from sequence, and we confirmed those by cutting DNA with commercially available enzymes that have the same specificities. And one of the restriction systems is shared between capricolum and mycoides. And the other 4 are presumably expressed when the donor DNA enters the recipient and would digest the caprecolum DNA. It was shown experimentally that these enzymes from mycoides digest caprecolum DNA. So here's what we imagine is going on now. The DNA gets into the cell. Again, still that part's a little mysterious. The enzymes are expressed and degrade the recipient chromosome, and that would account for the clean transplantation, not leaving any trace of the genetic material from the recipient. Okay, that's really all the results I'm going to talk about, but we're going to should I just wind this up? Okay, yeah. Don't ask me for this software. This is the slide that Craig uses, but doesn't exist. Thanks.